Okay, continuing with chapter 22, we're going to talk about the types of data that's actually used to document evolution. Now, the documentation of evolution has really started with fossils, but fossils are not the only types of data. And as we've spoke about before, that remember fossils are limited, right? So they're biased, right? So they're only the four types of data comes from what's called homology, and we're going to talk about homologous structures and analogous structures and this process called convergent evolution. Then we're going to talk a little bit more about fossil record. Biogeography is another area which provides some great documentation of the patterns of evolution and, of course, direct observations. Now we're continuing to discover new things that continue to fill these gaps that we might have had, say, in fossil record or any other types of data that have been obtained to I guess, document this pattern of evolution. Since, of course, Darwin wrote his Origin of Species, which was quite a long time ago. Homology is similarity between structures resulting from common ancestry or common descent. So these are structures that are very similar in organisms that all belong to the same lineage. So one arose from another, and another. An example would be these mammals, right? So mammals are a true monophyletic group. So this means they're all kind of related in some way. And they have structures that are related to each other as well. These are called homologous structures. So they're typically anatomical structures that resemble each of the organisms, even the ancestors prior to that. So this is immediately due to common descent. So an example would be these four limbs. Okay, so within the four limbs, the bones within the four limbs. So you can see they're kind of color coded, the ones that are matching in terms of similar structure and, of course, function. The only thing different here is that each of the mammals may or may not use the limb for the same purpose. For instance, humans and cats use their limbs to walk or run, right? Whales and bats are going to use their limbs to either swim or fly. But they all have the similar bones, and it's the bones, not the function of that particular limb. We're going to talk about the function of the limbs later, right? But it's the bones that are homologous. So they're similar based on common descent. Comparative embryology allows us to compare these anatomical features along the course of development of organisms. Now, just because there's a similar feature early on in development doesn't mean that that feature is going to remain there during the course of development. This is what the field of comparative embryology has identified. It is revealed that there are certain anatomical features that may be similar within a major group of organisms, for instance, the vertebrates, right? But not visible in the adult group once they go through their course of development. Now, the specific example given here is that all vertebrate embryos have a postanal tail during their course of development. So we can see the postanal tail. But we don't see that in our adult forms. We don't have a tail, right? So we don't have the postanal tail once we become adults. But this is a similarity among vertebrates in general. Now this kind of brings us to this other type of structure. It's called vestigial structures. Vestigial structures are actually remnants of features that served a function in the ancestors, and it could have been ancestors way back, right? So vestigial structures are just remaining components of it. So we might actually have that, like a partial part of that structure. However, it doesn't have a function anymore, right, in current organisms. Now, homologies allow us to create diagrams showing the relationships between organisms. These are evolutionary trees. So these evolutionary trees, and often called phylogenies, they're going to reflect the relationships of the organisms that we are studying. Now, these are just hypotheses, right? So it's just a hypothesis. This can be shown to be incorrect later on. And these evolutionary trees can be, or the relationships between organisms can be shown in the evolutionary trees using different types of data. It could be used using anatomical data or DNA sequence data, other types of molecular data. I must mention, though, anatomical data is not as reliable as molecular data. Now let's look at tree thinking in relation to homology only. This will be further discussed in chapter 26, and you will perform a lab at some point where you're going to be creating trees yourself. But I just wanted to explain how trees are made based on homology. So if we take a look at this tree, this is really just a tree showing just a small group of animals, starting with lungfish. If we take a look at this right here, number 1 indicates a branch point. So does number 2, number 3, number 4, number 5, and number 6. This branch point represents a divergence between a group of organisms, between two different groups of organisms. For this particular tree, the two groups of organisms that diverged at this point were the lungfish and this whole group right here, which includes what evolved much later, and that includes amphibians all the way to birds. If we take a look at the branch that precedes this branch point right here, this represents the ancestor of all the organisms within this entire tree. Now, if we consider homology, remember, homologous structures are structures that arise due to descent. Now, if we look at the first structure that's indicated on the tree, and there should be pretty much at least one structure indicated at every branch, but there's only 3 of them here, and we're only going to refer to these at this point. Digit-bearing limbs. So that means limbs with digits, and that evolved at this point right here. So of course, the lungfish do not have digit-bearing limbs. So if the lungfish did have digit-bearing limbs, then digit-bearing limbs would have occurred back here, so that this indicated that this branch point happened, and along the length after any particular structure that's indicated in the tree, each organism along that line that follows it should have that character. Now, that's not always the case, and that's kind of relating to convergent evolution. But convergent evolution is based on analogous structures, not homologous structures. So trees are best built using homologous structures. Analogous structures make it difficult to create a tree that's accurate. But anyway, let's say that all of these organisms that follow digit-bearing limbs on the tree have digit-bearing limbs. Now, they may not be the case. We have lizards and snakes that do not have digit-bearing limbs. But we'll refer to that at some point later on. So this would be a homologous trait, okay, for all of these organisms that follow it. The same thing would be true for the amnion and the feathers, except feathers is only a homologous trait that's shared between, in this particular tree, ostriches and the hawks and other birds. The amnion is a homologous trait that's shared among the mammals, the lizards and snakes, the crocodiles, the ostriches, and the hawks and other birds. So you can say that at this point, each one of these will be homologous characteristics, and that's how a tree is built. Now, convergent evolution is another concept which complicates the construction of an evolutionary tree. So convergent evolution is the evolution of similar features. However, they're similar features among distantly related organisms. So these organisms do not belong to the same lineage. This arises when groups independently adapt to their particular environment. So this could cause a problem or some more difficulty when constructing a phylogeny. Now, convergent evolution is not going to give any information about ancestry, right? Because these organisms are not related to one another. They just have a similar feature. So an example would be the sugar glider. Okay, so we have the sugar glider and we have the flying squirrel. They belong to completely separate groups. But look at them, they look very similar. They both have the ability to jump from tree to tree, almost fly, because they have that structure that looks like a wing in some way, right? So that's called an analogous feature, okay? Remember we talked about homologous. Homologous is similar based on ancestry. Analogous is similar not based on ancestry, right? So this is analogous features. This is just similarity in groups that have evolved in a similar way to adapt to their environment. Now let's see an example of a question. Look, I've got this example right here. We've got, suppose two species have a similar feature, and you are trying to decide whether the feature is homologous or convergent, or analogous would be the same term. What is the best strategy to use? So I'll give you a couple of minutes to try to make a choice, and then we'll move on to the next slide. Okay, so which one did you choose? You chose B, you are correct. So what you want to do is determine if their common ancestor also had the feature. So if their common ancestor also had the feature, what would it be? Would it be homologous or analogous? Remember, so the common ancestor suggests ancestry, common structure based on ancestry. So that would be a homologous structure. So let's look at the rest of these. Determine if the feature is vestigial. That really has nothing to do with determining whether an organism or a structure is homologous or analogous. Let's see. Determine if the feature is used for the same purpose, so the purpose doesn't matter, right? And also determine if the two species share other features. The other features have nothing to do with this one particular feature, right? So here we're trying to determine whether this one feature is homologous or analogous or convergent. And then A, determine if the feature is shared with a third species. That third species doesn't even matter, right? So that shouldn't have any effect on it. So what you want to determine is whether the ancestor has that feature or not. So we just talked about homology. So those are some ways to document evolution. Fossil record, you already know, is another one. So here's a little bit more example, right? So fossil record can provide provide data on extinction of species, when or about when new species had arose or new groups, and what types of changes occurred within these groups over time. So let's look at an example. We have the ankle bones of mammals and then comparing them to the cetaceans and the other and the even-toed ungulates. So these are kind of all related in some way. So we can see that the most mammals have they're almost equal in terms of these sizes. And then we have these structures right here. We have the cetaceans and then the other even-toed ungulates. So we can see these similarities, and then of course differences. Here we've got one side of this bone a lot larger than the other. Now, considering the possible limitations of the fossil record, we could look at this tree right here. This particular tree shows basically the length of time in which things diverged. So this one's a little bit different than the previous tree. And again, we'll talk a little bit more about this in chapter 26, but I just want to talk about fossil record and its relation to these organisms, the even-toed ungulates, the hippopotamus, and the living cetaceans. And then you can see in between here we have shorter branches as related to time, and this indicates at the end of the branch around, let's say, about 45 million years ago, this organism became extinct. Same thing with this one, about 40 million years ago, this organism became extinct. And about 35 million years ago, this one. But we have evidence of these in the fossil record. So if we did not have evidence of these in the fossil record, then these, we would not know how, or the relationship between living cetaceans and the ungulates. So we do have this, and we can see the progression of change from normal sized limbs to limbs which would serve better as ones for swimming. So this kind of shows the progression of the change in this type of in these ankle bones from organisms that evolved at one point, then through organisms that became extinct, all the way to organisms that exist now that are very closely related. However, they do. Now, biogeography is another form of data that can be used to document evolution. So if we study the distribution of species in terms of location, so where did we find these fossils, right? So what continents did we find these fossils? Remember, we talked about this before with continental drift. So remember, continental drift is this theory where, you have this plate tectonics, and we've got these movement of these plates, and it allowed the movement of the continents, and we can identify species that are very similar on totally different continents, which suggests that these continents were once together. It's important that we do understand the movement of the continents, and then of course the distribution of species that exist today, and then comparing this with what could have existed years from now, or years not ago, not from now. Now, it allows us to predict when and where different groups had evolved. And we could actually identify certain species that were not found anywhere else in the world. So if they were only found in one area, the term for this is called endemic. Endemic is a species, or an endemic species is a species that lived in one particular region. It wasn't found anywhere else in the world. It was pretty unique for that geographic region. So an example of biogeography, look, we've got islands of endemic species that are often found closely related to the species on the islands around it. Darwin suggested that species from the mainland, we could say, right, colonized the lands around it, so those islands around it, giving rise to new species because they've been separated. They're no longer, we'll talk about speciation later, but they're no longer mating with one another if they move to a different island and they, let's say they do not migrate. Okay, so biogeography can help us document evolution, really, the rise and fall of species. Now, direct observations is one that we often see today, but that's typically only with organisms that we can see rapidly, maybe rapidly evolving, but organisms that have a short generation time. So one example is of course, natural selection is the main source of this evolution that we can see, but we see this in the evolution of bacteria that become drug resistant, like MRSA. So MRSA, this shows the genome of MRSA. Look, we've got methicillin resistance. It's got a new gene that provides a product which allows it to survive in the presence of methicillin. So methicillin is no longer effective against this type of Staphylococcus aureus. Right, so we could new genes, so mutations can evolve, which result in new products, new genes, which can cause resistance. Now, methicillin is an antibiotic that works by inhibiting an enzyme that's actually used to produce the cell wall in bacteria. Now, this is kind of necessary. So if methicillin is working, then it keeps the cell wall from being produced so that the bacteria cannot propagate. Well, MRSA bacteria, these are the resistant ones, they use a different enzyme to produce their cell wall because they have that new gene, right? So this allows them to survive in the presence of methicillin because methicillin is ineffective, but because they have a different gene. They have a different enzyme that they actually use to produce their cell wall. So when MRSA strains are exposed to methicillin, they're more likely going to survive and reproduce. So then basically the only ones that are going to be surviving are going to be the MRSA strains or the resistant strains and overpower the strains that are more sensitive. Now, there's a number of new antibiotics that have been used. We have Teisobaxin, Bactin, which is a new one. But think about it, right? So let's say if we develop a new antibiotic and then we continually use that, do you think there will be resistance that could arise or evolve from using an antibiotic or overusing an antibiotic? Think about it. There's a good video that I want you to watch. Go ahead and you'll have to link to that separately. I'm not going to play that within this lecture. However, it's a really, really interesting video. So let's see a sample question, right? So here it says MRSA infections are occurring at alarming rates. One reason for this could be that people do not finish their antibiotics. Right, so this is a problem. You might have thought about that when I told you to kind of think about it in the previous slide. Which of the following is the most likely reason that could lead to something like MRSA? Right, so let's look at this. We've got A, antibiotics take some time to start working, giving the bacteria time to mount defenses against the antibiotics over a few days. So think about it. Do the bacteria mount defenses? Now here we have B. Because antibiotics are often taken where there is no bacterial infection, the antibiotics aren't using their medicinal antibacterial properties and are losing them. So are the antibiotics losing their medicinal properties? And then the third one says, the first few days of antibiotics kill off the susceptible bacteria, making people feel better. So then often people that feel better, they stop taking their antibiotics. But then it's not killing all of the bacteria, so some of the bacteria are still surviving. So when people stop taking that antibiotics, the resistant bacteria that have survived this treatment can reproduce and create more resistant population. So which one do you think it is? Are correct. All right, now one final thought on natural selection. So natural selection can actually respond to new species, new environments, right? So let's see here. We've got the soapberry bugs. They use their beak to feed on the seeds within the fruits, and we find these in Florida. So feeding is most effective when the beak length matches its seed depth within the fruit. So what this means is that if the fruit have a deeper seed coat, right, so the fruit is deeper inside the seed, then the longer beak would be more helpful. So a longer beak wouldn't really evolve if the seed depth was not as long, right? So if it was shorter. we've got South Florida and Central Florida soapberry bugs. So in South Florida, these are larger fruits. So the soapberry bugs typically feed on these balloon vine, right? So they have larger fruit. So the soapberry bugs have evolved to have longer beaks because the larger fruit are going to have a larger seed coat. So the depth of the fruit is going to be a lot larger. In Central Florida, they're generally going to feed on the golden braid tree. These have smaller fruit, so they have smaller beaks. So they basically, you can see this. We can actually, this is a direct observation. We can see natural selection. We can see this evolution happening, right? So in South Florida, larger fruit, right? Deeper fruit. They need to actually be able to go deeper into the seed to get to the fruit. So they need longer beaks. So that's going to evolve. So those with longer beaks are more likely going to survive and reproduce because they can feed off of the fruit. And then you're going to see more quantity of the longer beaks in that particular area. Same thing with the Central Florida. Let's say it started out with short beaks. They didn't need to evolve longer beaks because the shorter beaks were working. So now you're going to see more shorter beaks because, of course, they do not need to evolve the longer beak. So there's a correlation between fruit size and beak size. And this is also seen in other states as well. So it's not just Florida. In summary, evolution is supported by all types of scientific evidence, and this evidence has been documented in these types of data. Homology, homologous and analogous structures. Basically, homology allows us to document evolution in terms of creating a phylogenetic tree or evolutionary tree because its similarities based on descent. And then fossil record, of course, we've covered fossil record quite a couple different times. Biogeography, remember that's tracking of continental drift and being able to identify fossils and species that have lived in any particular geographical area. And then direct observations, we see the effects of resistance to antibiotics and the example given with the soapberry bugs. All right, so that concludes chapter 22. Thank you for listening.